Thank you, Ethan. Yeah, today I'm very pleased to share the news about two tyrosine kinase inhibitors. The first one is Gleevec, and it was developed by Novartis. And in fact, this study report published at the New England Journal of Medicine on April 5 was immediately followed by the drug approval in May of 2001. And at the same month, it was also on the cover of Time magazine. I still remember the ASCO that year happening at San Francisco, and I squeezed in to this ASCO. Mainly, there was the Herceptin story, but the timing was just perfect for us to realize there is the birth of targeted therapy in the form of antibody as well as a small molecule. So the reason I started with this molecule Gleevec is because it is an inhibitor for the target BCR-ABL found in chronic myeloid leukemia or CML patients. This fusion protein caused the uninhibited activation of the ABL kinase because of the translocation of the BCR protein to the N-terminus. So next slide. As you can see, the timeline on the left side, in the CML drug development, the first decade was marked by the imatinib, the Gleevec molecule that really transformed the CML landscape from a fatal disease into a chronic lifelong condition. People can live the same lifespan, and the response rate is over 90%. But gradually, people develop resistance. That's why in the second decade, in 2010s, you started to see second generation active site tyrosine kinase inhibitors. And on the chart, on the right-hand side, you can see this second generation have better efficacy, but less desirable in terms of safety and tolerability. So now, up to 2020, we started to see the allosteric inhibitors represented first by, again, the molecule from Novartis, Asciminib. And this molecule has better efficacy and better safety. Now comes TERN-701, and based on the data, it claims to have the potential to be the best-in-class allosteric inhibitor. Next slide. So what is allosteric inhibitor of BCR-ABL? This figure comes from the New England Journal publication for Asciminib, and it shows that on the left side, the wild-type ABL1 kinase has N-terminal myristoylated module myristylation in N-terminal, followed by SH3 domain, SH2 domain, and then followed by kinase domain. And in normal circumstances, the myristoylated N-terminal plugs into the kinase, make it a folded conformation, and the kinase is not active. And when you have the Philadelphia chromosome translocation, BCR protein displaced the myristoylated N-terminus. Now you leave the protein in the open conformation, and the kinase is constitutively active, and that drives the oncogenesis. And when you have an allosteric inhibitor, a small molecule such as Asciminib will plug itself into the myristoylated pocket and in a way reconstitute the inactive form. And as you can imagine, allosteric inhibitors of BCR-ABL can target both the native as well as mutated BCR-ABL1, and including the gatekeeper T31I mutant. Additionally, because it does not inhibit the wild type ABL1 or ABL2, then this combined unique MOA gives them both efficacy and safety advantages. Next slide. So TERN-701 was internally discovered by Terns Pharma, and which was founded in April 2017. So 2 years later, they discovered TERN-701 and nominated the candidate. And then in 2020, Hansoh in-licensed the China right, Greater China right, including China, Taiwan, Hong Kong, and Macau, from Terns, and it was very much in line with the trend of how China's biotech developed over the last decade. And there was an interest in licensing the molecules and assets from Western world and develop them in China. And indeed, Hansoh started the phase 1 trial in 2022. But subsequently, Terns also started its own internal program, including a Western healthy volunteer single ascending dose study, then followed by phase 1 studies in previously treated CML patients. Next slide. Next slide, I think it shows Hansoh's study design. Also in the CML patient with Philadelphia chromosome translocation and who are resistant or intolerant to active site TKIs. They started with 40 mg and went as high as 320 mg, and this is once daily dose. With this, I initially thought it probably contributed to the Terns' own effort, but turns out Terns' healthy volunteer study started with 20 mg. It probably helps Terns' CML patients trial. Next slide. The CARDINAL trial is in this month's news. It started from 160 milligram and went as high as 500 milligram once daily. And subsequently, 2 doses were taken into the expansion cohort, 500 milligram as well as 320 milligram. The results right now only showed the first 32 efficacy evaluable patients in the dose escalation portion. Next slide. Maybe some in the dose expansion portion. And the way they assess the phase 1 activity is by the molecular response, which measures the transcript of BCR-ABL in peripheral blood by quantitative PCR. So if your baseline is 100% level of BCR-ABL, if you reduce it by 1,000-fold such that the level is below 0.1%, this is called major molecular response, MMR. And if you go down further in over 10,000 and above kind of a reduction, you get a deep molecular response or DMR. So the assessment for CARDINAL study was based on safety, tolerability, as well as the major molecular response. Next slide. And they compared their MMR rate and called it an unprecedented 64% MMR achieved. This is in comparison with Asciminib, which is an allosteric inhibitor in phase 1 with 99 patients, triple the number of patients so far against the TERN-701. It showed 24% MMR achievement at 24 weeks. And even in the phase 3, it shows 25%. So Terns, even though it has a smaller data set, and even though this is cross-trial comparison, it shows a promising signal with 64% MMR achievement. And again, I did not show all the patients' treatment background because these patients have experienced the first generation, second generation, even the allosteric inhibitor of BCR-ABL, and being able to respond so well to TERN-701, and that's very impressive. And also at all those levels, they do not see those limiting toxicity. So the safety profile is very impressive. Next slide. As a consequence, Terns stock is almost tripled. It's from November 3rd when their abstract at this year's ASH next week is published. The stock went to 250% up. And interestingly, this program seems to be on the quiet side until now. So Terns got its new CEO, who is a friend of mine, Amy Burrows, who used to work in a commercial group in Genentech. Terns is Amy's second CEO duty. She folded the first company where she was a CEO at Cleve, and then took this public company. And during the last year, she joined in February 2024. Last year, a lot of activity seems to be on the cardiometabolic or obesity. Actually, it's the obesity side of Terns Pharma. But the data readout was disappointing. That just happened in October. Interestingly, there was not a strong stock reaction. And also for the management, there was not any sign of maybe there's a disappointment, but the management has a plan to go ahead to look at a different program, which is in blood cancer. That's really totally different from obesity. But if your abstract is published on November 3 for ASH, you have to submit your abstract in August. So that means they probably know the data in August already, and that's what they are focusing on. And they really have done a fantastic job telling their stories of the TERN-701 program, and they also decided they did not want to raise upon good data, but it is really good to see the stock market is coming back to normal in that good data are being appreciated and recognized. I think I will stop here. I think if you have one more click. It's a good segue to my next story, but let me pause to see if people have any questions.
Yeah, right now there's no question. You can just proceed. Any question in the chat, I will bring this to you.
Okay, yeah, so I started with the first direct kinase inhibitor. Go back a little bit. Which was Gleevec, right? This is if you don't consider rapamycin as 1997 approval. In this paper that I showed the link here. This is to celebrate 100th kinase inhibitor being approved by the FDA, and Gleevec being the first one, or number two if you count rapamycin, in 2001, and now the 100th molecule is actually a BTK inhibitor. It's called remibrutinib also from Novartis. And so now my second piece of news this month is about BTK inhibitors.